Okay, so today we're going to talk about the notion of a functional state. We have discussed this before, but it's important to go through this concept in a little more detail because it is a foundational one in systems biology and something that you may or may not learn from biology classes. Maybe in human physiology, if you take a class on human physiology, circulation, and so forth, you get to appreciate the functional state a little better. But we're going to talk about that in more detail here in this lecture. So the outline is as follows. I want to talk a little bit about the view of components versus the view of a system. Most of the classes we take in biochemistry or molecular biology is focused on components, but systems biology looks at the system, and I want to bring a little more contrast into that. I want to talk about links in a network that we know, of course, to be ultimately to be chemical transformations or chemical transactions amongst molecules, but we always don't have that level of resolution of detail when it comes to many cellular functions. I think we have seen how links or individual reactions lead to networks or network reconstruction, but having a network leads to the possibility of computing a functional state. And that's kind of the focus of this lecture. We'll talk a little bit more about the notion of constraints and how they eliminate certain functional states and allow them. And at the end, I'll say a few things that are maybe a little more broad perspective. It's about the biological consequences of constraints and the fact that, much biological phenomena we observe as a consequence of abiding by constraints. Okay, so first up, the thinking in molecular or component biology versus systems biology. So here's a typical image of a protein or a multimeric protein that may carry out a reaction. You may constitute this protein in a beaker, put the substrates in there, and watch the reaction unfold. And your focus is going to be on that enzyme and its activities, right pH, need for cofactors, and so forth. But once that enzyme is put in the context of a network, it is not operating independently. It has to abide by what its neighbors are doing in the network and the fact that the cell needs to attain some sort of a homeostatic state. So all the components together have to exhibit some coherent function. And in fact, the need to have a balanced flux map is actually the primary concern of a cell, and the function of the components has to be according to that. So you need the right abundance of a component times its activity to get its overall activity in a cell, and that has to balance with its needs in the network. And we all realize how this goes in human societies and networks. There's a bunch of things always pushing us around to do things that we don't really want to do, but we do them to make sure everything functions in harmony. Okay, so we can observe components, or we can evaluate them in isolation, that is, or evaluate their function in the context of everything else that happens in the cell. And when you start thinking that way, your focus shifts a little bit towards thinking about systems and the need for networks as a whole to function. So we'll first talk a little bit about links. I think we know that essentially all chemistry that takes place in cells is facilitated by proteins. And most chemical reactions actually happen on the surface of a protein. And the enzyme determines the value of the rate constant. I'll talk about that here a little bit. So if two molecules are bouncing around in solution, A and B, they may be colliding off one another, and if they happen to hit each other at the right angle and at the right with the right momentum, you will see a reaction happen. And that's maybe the natural rate of reaction. But an enzyme will bind these components first to a surface and put them in perfect juxtaposition to each other so that the likelihood of a reaction taking place is increased enormously. And many of us have seen these studies of million-fold acceleration rates by enzymes. So if these collisions are unlikely, low frequency, or the angle of collision is stringent, the enzyme basically knows that and puts the enzymes in the right, the substrates in the right position relative to one another. So that actually means two things. So there is information in the surface of the enzyme about the reaction. And that information ultimately is encoded in the genome and is based on the history of the organism. It has learned how to do this reaction at the right rate by designing that protein or selecting for that protein or protein properties accordingly. So there is memory in the genes, and that's reflected in part by these very special properties of a surface of a protein. But it also tells you another thing that's very important, which is that the rate constant is a biological variable. Changing the surface will change the rate constant. It won't change the stoichiometry of the reaction. It will not change the thermodynamics either. Those are physical chemical properties that are fixed. But the rate constant is malleable and is adaptable biologically. And a lot of designs of networks comes down to having the right set of rate constants selected for. Because if any one of them is out of balance, the whole network may not function well. It has to be fine-tuned and work perfectly. So that's where the biological factor really comes into determining the properties of links. Stoichiometry may fundamentally exist, but how do you make that reaction function inside the cell? So for some cases, this has reached an incredible level of sophistication. And this example here is the Rna Polymerase. That is maybe one of the more complicated enzymes in the cell. It latches onto the DNA, opens it up, pulls the strands in a certain way on the surface, and makes a new RNA transcript form. And then when it falls off, the DNA closes again. All of this is a function of the surface properties and certain dynamical properties of this very complicated complex of protein that come together. So this may be an extreme case, but for most metabolic enzymes that we're going to be talking about, this is simpler, but the principle is the same. There is surface properties of the protein, right prosthetic groups, right geometry, and so forth that makes the reactions happen at the rate that the cell needs. Okay, so those are a few things about protein. So let's talk a little bit about chemical reactions as links in a network. So here is an image that shows you how much information we have about this process. To the right, we have an arrow that is perfectly in focus. That's when we know everything. Our understanding is clear and sharp. This is when we know the kinetics, we know the thermodynamics, and we know the stoichiometry. Maybe one step short of that, which is where we're going to operate, is where we know the chemistry, we know the structure, we may know the thermodynamics, but we certainly don't know the kinetics on a genome scale. So from a practical standpoint, at this level, the chemistry may help us quite a bit, but helps us a lot actually, but the arrows may be a little bit blurry. If you go further to the left, these relationships become more and more abstract, and as you know, in some cases, the best you can do is to do some sort of a correlation between two components, whether they are there or not in all functional states of a cell. Sometimes you get causation by knocking out the gene and so forth, but down at that end of the spectrum here, your understanding is more blurry, it's less precise. And of course, it's maybe where the opportunity for discovery is much higher. So there's a spectrum of the understanding of links in networks inside cells, and that's what this illustrates. Okay, so once you put a network together with all these links, you have a complicated knowledge base, as we talked about, the reconstruction. So the reconstruction has this network or an assembly of all these chemical transactions, and we now like to put them into a mathematical format and compute what the state of the network is. So let's talk a little bit about what a functional state is. So we live in networks all day long. Traffic, coming here in the morning, placing a call on your cell phone, plugging the computer into the wall, power grid, a lot of networks all around us. So here's one. This is the highway map of Los Angeles, and this is a network, and it has a certain topology and is always there. This one we actually know fairly well. But what we may not know is what its functional state is from time to time. And of course now we can go online and we can look at these traffic patterns at different parts of the day, and they will always be different. So the activity state of that network, its functional state is different, even if the network is always the same. And that's a very important concept. So as another biological example, the basic metabolic pathways in your liver are in a very different functional state when you're fasting versus when you're fed, but it's the same network. And if you're fed with a fatty diet or carbohydrates or protein, the functional state will always also be different. So this is very important, that biological networks can have many, many different functional states. And those are the ones we want to compute once we have the topology or the structure of a network. So here's an example from the literature. This is from Uwe Sauer's laboratory at the ETH, where he published his paper studying the activity state of the basic pathways in E. coli. And on your left, you see basically what's in our core E. coli model, the end of glycolysis in the TCA cycle, and this is very consistent with what we read in our biochemistry textbook. So a lot of glucose flowing down into the TCA cycle making ATP. But on your right, there's another functional state of that system, which is closer to a starvation state for E. coli, where it's limping along. And it uses exactly the same reactions, but with different flux loads, and the pathways are doing slightly different things in terms of the net overall activity in the state on the left versus the one on the right. So this is very real, and we can measure these states sometimes, and we certainly would like to be able to compute them. So how do we compute them? The process we go through is the one we've been talking about. You have a real cell, you have information about all its components, you build that network, you formulate a model, and now you have some in silico version of your cell that is consistent with everything you know about that cell. Is it exactly the same thing as the real cell? It obviously is not, but it hopefully will have many properties that are the same as those of the real cell. So here's the process that we're going to be going through in part 3 of this class, building the models. So in part 2, we saw the fact that solution spaces associated with the network equations are finite, and any point in that state in that space is a candidate state of the network, and these are all the allowable functional states now. And what we're going to be doing here is to use optimization methods to look for particular points in that space. Now, how do we think of functional states? We may not always think of them from the same point of view as you see in a textbook. And to the top right is, again, that image we use of core metabolism in E. coli. It is the usual pathway map, the primary pathways and how they fit together. But if you start looking at S transpose, then you start looking at the function of the cofactor pools in there that are so fundamental to having a network function correctly. You may take a different view of that network, and that's the big image shown in this diagram here. So on the right, there is the extraction of the redox equivalents. They may be put onto a number of different redox carriers shown in that center column. And then the uses to the right could either be, towards the bottom, the delivery of the redox potential in a substrate onto the ultimate recipient of that through biosynthesis, say, or you can use the redox potential to pump protons, and translocation of protons across a membrane can drive the ATPase. So this is more like a conveyor belt. This is a bunch of cycles. This is how properties in metabolism are cycled over and over again. If you start looking at signaling pathways and other functions inside cells, when you take more of a bioengineering view or a systems view, you may start to think about networks in this sense, because this is more functional than what you may have up in the top right corner. Both, of course, are useful. So the systems viewpoint, at least in this case, may lead you to viewing the transmission and trafficking of redox potential as a tangle of cycles, because of these molecules constantly going between an active and an inactive state. Okay, so just a little additional comments about how we think about functional states there. So let's talk about the constraint-based thinking. So this is something I mentioned a little bit last time, but we'll go through it a little bit more here. So to the left is the sort of thing you may see in a physics class or a chemistry class or an engineering class, where you're asked to calculate a solution. And there is a correct solution, which is represented by that red dot. And often if you take the test and you get close, you may get partial credit. And I, as a professor of engineering, used to grade exams and try to figure out how close to the right answer people were and grade them accordingly. But what you see on the right is more the thinking that we see in systems biology, where there's a whole space of acceptable solutions. And there are many of them, and they're not unique. And we need to study them as a group and try to determine their properties and try to determine which one of these states are biologically meaningful and which of them actually give optimal function of an organism through evolution. So this leads to this interesting table here that contrasts a modeling philosophy in different disciplines. So if you take a class in physics, it's all about the equations. It's all about figuring out what the laws of nature are. If you want to solve them, you state the initial conditions. But that's not so important. We need to understand the theory and the equations. In engineering, you need the equations. They may not be fundamental, they may be phenomenological, but you need them to quantify things. But for an engineer, the boundary conditions and the specific conditions are extremely important because they represent the design that you're after, as an example. It appears when you start thinking about biology that through the constraint-based modeling, the ISO constraint-based modeling, is that the equations will only take you so far. But the conditions are very important because they set the boundaries, they set the constraints and so forth. So the equations, as long as they are consistent with fundamental properties that we can represent mathematically easily with minimal amount of information, that's maybe sufficient, but we may need a lot of information about the conditions under which we're studying the cell, because they're always context dependent in their function, and we can generate a lot of data on that and figure out the boundary conditions, the constraints that are on a network of interest. So, as we've talked about several times, the constraint-based field started in the late 1990s. First genome scale models came out around 2000. And within a few years, there were a lot of constraint-based methods developed. There's about a dozen of them on this diagram here. And today, we have many, many more of them. And this review was prepared by Nate Lewis a few years ago, where he found over 100 different methods that are executed under the constraint-based paradigm. But they fall into categories. So this was described by a phylogenetic tree. So there's a group of methods that allow you to assess the consequences of losing gene function. There is a group of methods that allows you to map omics data. There's a group of methods that allow you to do gap filling, figure out inconsistencies, and so forth. So Nate will be here on Tuesday to go through this phylogenetic tree. And he maintains a website that should be on here. I guess it's not on here, but he'll show you where it is, where he tries to post every new one of these methods. And I think he says there are about 200 of them. But some of them may be becoming a little more finer nuance of a set of methods that already exist, so there may not be that much anymore that's radically new and different. But there are always some things happening. And you can go to the COBRA conference if you're interested to learn about the latest and greatest in the field. Okay. This slide we've talked about, so we're going to skip it. So let me talk a little bit here at the end about constraints, not so much in the sense of trying to compute the phenotype in a given conditions, given a lot of data, but from a broader context, from a biological context. So first, we have talked about this diagram here that interprets regulation as a willful way of shutting down candidate states, that the cell somehow by its evolutionary history knows are not good in a given condition. So it downregulates genes, implements new regulatory mechanisms to prevent those from happening, and drives itself into a part of that solution space where it wants to operate. So that's one biological consequence of constraint-based thinking. Regulation becomes a set of restraints for the cell to get to its functional state. So here's another one. So we know that DNA is hierarchically organized. The length of the DNA molecule, for instance, in E. coli is 1,000 times the length of the cell. So it has to be folded up 1,000 times. And there's a lot of constraints on how you do that, and then how you unfold it, how you access it, and so forth. And this leads to a very definite hierarchical organization of the genome in space. And the same thing is true of the human genome. I think it's like 4 to 5 feet in linear length, and it all has to be packaged up into the nucleus in the form of all these chromosomes, and they have to open up and close and so forth. And there's also a very well-defined hierarchy of how DNA is organized spatially inside cells. So that's basically a result of all sorts of constraints. It's a linear molecule, you have packing constraints, and you have to be able to access the genes in a particular way. The same thing is true with network function, because at the top end, well, bottom of this slide, at the high end of this hierarchy, you have to have a cell that is functioning. You have all these components down there, and there's a lot of constraints on how they have to function, so they start to modularize to form a coherent function that successively becomes bigger and bigger modules. So hierarchical organizations in biology is really one consequence of all these constraints that are placed on biological function that we've talked about. And so here is slide that I've, a quote that I've shown you, I think, before from Sir Paul Nurse, wrote a very perceptive piece in Nature that is cited there after he got the Nobel Prize. And down there is illustration of the hierarchy in biology from molecules to human societies to, I don't know if ecology is on there or population level. At each one of these levels of this organization, there are different constraints at work. And when one level kind of operates under full constraints, it may evolve onto creating a higher level function under all these constraints. And then it run into another set of constraints and so forth. And that's illustrated by this diagram that came from Mark Kirshner at Harvard, who started the first systems biology department. And here he talks about successive fixation of biochemical constraints with evolution. So for instance, early on, the basic metabolic processes evolved, and DNA as a storage molecule was picked. Those are now more or less fixed parameters in all the evolution that occurred after that. You're not going to change your storage molecule overnight. That's already been decided. The glycolysis is already in use. And then, of course, eukaryotic cells, they have functions that, like the mitochondria, that originates in prokaryotic life, and that sets constraints on eukaryotic function, and so forth. The generation of multicellularity, the body plan, the limbs, and so forth. So evolution over time, as we see evolution of more and more complexity, builds on processes that have already been evolved to fit certain constraints. So constraints-based thinking really permeates biology when you start looking at it carefully. Okay, so a few summary bullet points from this relatively short lecture. So biological systems at the molecular level are defined by interactions between their components. The links that are formed between components obey the basic laws of chemistry, and we talked about a few of these. Once you have multiple links operating, multiple reactions operating, you stop thinking about the individual reactions, and you start thinking about a network. So a network has the function, pathway may be, maybe not quite a network, but it's a coordinated function of multiple links. Multiple pathways form a network. And then networks as a whole exhibit coherent function that ultimately is a homeostatic state that we refer to in systems biology as a functional state of a network. Now, allowable functional states are heavily constrained by a number of factors that we have talked about. The environment, basic physical chemical constraints, and so forth. Now, it turns out that, and we mentioned this a little bit in the lecture on pathways, as a network becomes more complicated, the number of candidate functional states grows very fast. And for instance, the extreme pathways that represent vertices in a polytope, once you get to 100 to 200 components, you may have thousands of those. And then if you get to a genome scale network of, say, hundreds of reactions, multiple hundreds of reactions, the number of these vertices may be in the millions, if not in the billions. So network topology allows for many, many functional states, but they are many, many more than are useful to the organism. So only a subset of these extreme states or close to extreme states in cells actually get selected for and get used biologically. And therefore, we have to have regulatory mechanisms to eliminate the undesirable states and close the solution space around these states that are of desirable properties. Now, we have a series of these constraint-based methods available to us now that enable us to study models from this standpoint, and we will be talking about them for the next few lectures. And when you start looking more broadly at biological functions, evolution, their organization in space and time and so forth, you start realizing that most of those structures and most of those events can be interpreted from a constraint standpoint. And for instance, we talked about tumor formation a couple of lectures ago, which is really a serial alleviation of constraints, and opens up the solution space into behaviors that are highly undesirable for the organism as a whole. So that's the end. The lecture is really aimed at trying to illustrate what is a functional state, and there are many of them that are consistent with all the constraints we impose on a system, but only a subset of those are useful. And there is some optimization criteria for picking the right functional states, and those selections have presumably been done by the organism by its evolutionary history.